Hello students and welcome to Learno Biology. My name is Nandini and in this video we will be discussing glycolysis process. So let's start the video. Before moving on to the actual steps of glycolysis, we need to understand what does glycolysis means. Glyco का मतलब होता है glucose and lysis means breakdown. So glycolysis is a metabolic pathway where glucose, a 6-carbon molecule, is broken down into pyruvate, which is a 3-carbon molecule. This multi-step process yields 2 molecules of ATP, 2 pyruvate molecules and 2 high-energy electron-carrying molecules of NADH, along with 2 molecules of water. In this process, glucose is broken down into 2 molecules of pyruvate. Moving on, glycolysis takes place in cytoplasm or cytosol of the cell. Glycolysis एक 10-step process है which is catalyzed by 10 specific enzymes. Glycolysis can occur with or without oxygen. If glycolysis is taking place in presence of oxygen, we call it aerobic glycolysis, and if in case the glycolysis is taking place in absence of oxygen, we will call it anaerobic glycolysis. The purpose of this process, glycolysis, is to release energy in the form of ATP. The whole process of glycolysis can be divided into 2 phases. First phase, where glucose is converted into glyceraldehyde 3-phosphate. This phase actually consumes ATP, hence this is the energy consumption phase. Talking about the next phase or the second phase of glycolysis, where glyceraldehyde 3-phosphate is converted into pyruvate, this phase actually harvests energy. In short, glycolysis is the metabolic pathway where glucose is broken down into 2 molecules of pyruvate to release energy in the form of ATP. Now let us look at the steps of glycolysis. This is the very first step of glycolysis where glucose is converted into glucose 6-phosphate. One phosphate group is attached to the 6th carbon of glucose ring, and this reaction is catalyzed by enzyme hexokinase. Talking about hexokinase, hexo means 6, denoting 6-carbon compound, that is glucose, and kinases are the group of enzyme that transfers one phosphate group to the other compound. Here, one phosphate group from ATP is transferred to glucose 6-phosphate and ATP gets converted into ADP. The enzyme hexokinase phosphorylates or add a phosphate group to glucose in cell's cytoplasm. So this process is taking place in the cytosol of the cell. In this process, a phosphate group from ATP is transferred to glucose producing glucose 6-phosphate or G6P. One molecule of ATP is consumed in this process. As mentioned earlier also, that in first phase of glycolysis, energy is actually consumed. So here glucose is converted into glucose 6-phosphate with the help of hexokinase enzyme, and here one ATP is used and it gets converted into ADP. Now in the next step, glucose 6-phosphate is converted into fructose 6-phosphate, and this reaction is catalyzed by phosphoglucomutase. This is just a rearrangement reaction. Glucose 6-phosphate and fructose 6-phosphate are isomers. Isomers have same molecular formula as each other but different atomic arrangement. So glucose 6-phosphate convert हो जाता है into fructose 6-phosphate. After this, in the next step, fructose 6-phosphate is converted into fructose 1,6-bisphosphate after the addition of one more phosphate group to the first carbon of fructose ring. This reaction is catalyzed by phosphofructokinase. Again, kinases are the group of enzymes that help in the addition of phosphate group to any compound. Here also one phosphate group is attached to fructose, converting this into fructose 1,6-bisphosphate, and ATP gets converted into ADP. Phosphofructokinase uses another ATP molecule to transfer a phosphate group to fructose 6-phosphate in order to form fructose 1,6-bisphosphate. 2 ATP molecules have been used so far. So अभी तक ATP के 2 molecule use हुए हैं. First ATP was used when glucose was converted into glucose 6-phosphate, and now the second ATP is used where fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Moving on to the next step, fructose 1,6-bisphosphate splits into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This is the product of aldol condensation reaction, and the enzyme that catalyzes this reaction is aldolase. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are isomers. The enzyme triose isomerase can convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, as glyceraldehyde 3-phosphate is the substrate that is needed for the next step of glycolysis. So dihydroxyacetone gets isomerized into glyceraldehyde 3-phosphate. Now we have 2 molecules of glyceraldehyde 3-phosphate. Next step is very important. The enzyme glyceraldehyde 3-phosphate dehydrogenase plays a very important role, and it serves 2 functions in this reaction. First, it dehydrogenates glyceraldehyde 3-phosphate by transferring one of its hydrogen molecule to the oxidizing agent nicotinamide adenine dinucleotide, that is NAD, to form NADH. This NADH is used in the process of Krebs cycle and electron transport chain to produce more energy. In short, in this reaction, one hydrogen molecule is removed from glyceraldehyde 3-phosphate and added to NAD to form NADH, which is used in the process of Krebs cycle and electron transport chain to produce more and more ATP. Next, glyceraldehyde 3-phosphate dehydrogenase adds an inorganic phosphate present in the cytoplasm of the cell to oxidize glyceraldehyde phosphate or GAP to form 1,3-bisphosphoglycerate. Now, 2 phosphate groups are added to this glyceraldehyde molecule. Both molecules of glyceraldehyde 3-phosphate produced in the previous step undergo this process of dehydrogenation and phosphorylation. Please remember, we got 2 molecules of glyceraldehyde 3-phosphate as dihydroxyacetone phosphate was converted into glyceraldehyde 3-phosphate with the help of triose phosphate isomerase enzyme. So as you can see it over here, glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, and this is the product of oxidation reduction reaction. Along with oxidation reduction reaction, phosphorylation is also taking place as 2 inorganic phosphate present in the cytoplasm of the cells are added to glyceraldehyde 3-phosphate to convert it into 1,3-bisphosphoglycerate. Here NAD is converted into NADH, and the whole process is catalyzed by glyceraldehyde 3-phosphate dehydrogenase enzyme. In the next step, enzyme phosphoglycerokinase transfers a phosphate from 1,3-bisphosphoglycerate to a molecule of ADP to form ATP. इस process में ATP का production हो रहा है. This happens to each molecule of 1,3-bisphosphoglycerate. As we were having 2 molecules of 1,3-bisphosphoglycerate, so this will happen to each molecule of 1,3-bisphosphoglycerate, and 2 molecules of ATP will be produced by the end of this step. This reaction yields 2,3-phosphoglycerate and 2 molecules of ATP. In simple word, one phosphate group from 1,3-bisphosphoglycerate is removed and this is added to ADP to form ATP, and after this 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. Next step is simple isomerization reaction, where phosphate group is relocated from 3rd carbon to the 2nd carbon of phosphoglycerate, where 3-phosphoglycerate is converted into 2-phosphoglycerate, and this reaction is catalyzed by phosphoglyceromutase. Moving on to the next step, the enzyme enolase removes a water molecule from 2-phosphoglycerate to form phosphoenol pyruvate. So one molecule of water is removed from phosphoglycerate to form phosphoenol pyruvate, and this reaction is catalyzed by enolase enzyme. And by the end of this reaction, we will be getting 2 molecules of phosphoenol pyruvate. In the last step, enzyme pyruvate kinase transfers a phosphate group from phosphoenol pyruvate to ADP, and ADP gets converted into ATP. So we will be getting 2 ATPs over here, as 2 phosphoenol pyruvates are there in the reaction and 2 phosphate groups are being removed. So 2 ADP will be converted into 2 ATPs. And by the end of this reaction, we will be left with 2 molecules of pyruvate. So in the second phase of glycolysis, we got 4 ATP. If we talk about the whole process of glycolysis, in the first phase 2 ATPs were used, and in the second phase 4 ATPs are produced. So in total, we got 2 ATP. This is the flow chart or the whole process of glycolysis. That's it for this video. If you have any doubt, you can put your doubts in the comment box. If you like this video, please subscribe to Learno Biology, and for more videos, keep on watching Learno Biology.